Hello friends, today we will learn about innate immunity. This is the second video of innate immunity. So let's begin with determinants of innate immunity. The main determinant of innate immunity are genetically controlled, varying widely with species and strain and to lesser extent between individuals. Age, sex and hormone balance play lesser roles. First point, we'll study about species and strain. Different species exhibit marked differences in the susceptibility to infective agents. Example, the rat is strikingly susceptible to diphtheria, while the guinea pig and man are highly susceptible. The rabbit is particularly susceptible to myxomatosis and man to syphilis, leprosy, and meningococcal meningitis. Within a species, different races may show differences in susceptibility to infections. This is known as racial immunity. Example, American Indians and Negroes are more susceptible to tuberculosis than Caucasians. Genetic resistance to Plasmodium falciparum is seen in some parts of Africa and Mediterranean coast. The differences in innate immunity exhibited by different individuals in a race is known as individual immunity. Example, heterozygous twins exhibit different degree of resistance to tuberculosis and leprosy, whereas homozygous twins exhibit similar degree of resistance. High susceptibility to infectious diseases is encountered in the two extremes of life as compared to adults. Some pathogens cross the placental barrier and cause overwhelming infections resulting in fetal death. Example, rubella, cytomegaloviruses and toxoplasma gondii, which leads to con- congenital malformations. Newborn animals are more susceptible to experimental infections in suckling mice but not in adults. The heightened susceptibility of fetuses and young ones is related to the immaturity of their immune apparatus. In the elderly, on the other hand, physical abnormalities, example, prostatic enlargement leading to stasis of urine, are a common cause of increased susceptibility to infection. Nutritional influences. Let's know about the nutritional influences. Experimental evidence in animals has shown repeatedly that inadequate diet may be correlated with increased susceptibility to a variety of bacterial diseases, and this has been associated with decreased phagocytic activity and leukopenia. In case of viruses which depend upon the normal metabolic function of the host cells, malnutrition, if it interfered with such activities, would be expected to hinder proliferation of the potentially ineffective infective agent. There is experimental evidence in support of this view in a number of animal species, which, when undernourished, were less susceptible to a variety of viruses, including vaccine virus and certain neutrophic viruses. The parasite requires para-amino benzoic acid for multiplication, and this may be deficient when a low level of nutrition exists. Now let's know about mechanism of innate immunity. Mechanical barriers and surface secretion. Let's know about the mechanical barriers and surface secretions. Most of infections that are individual encountered do not penetrate the body surface but are prevented from entering by a variety of biochemical and physical barriers. The body tolerates a number of commensal organisms which compete effectively with many potential pathogens. First, let's know about the skin. First point is horny layer, that is stratum corneum, a mechanical barrier. Second point is lysozyme of sweet kills gram positive bacteria. Third point, desquamation, desquamation, that means loss of superficial layer with attached microorganisms. Fourth point is sweat and sebaceous secretions, lactic acid and fatty acid, lower pH of skin. Fifth point is skin microflora. Example, propionobacterium acne produces antibacterial fatty acids. Next point is second point is mucous membranes. Cilia of respiratory tracts and coughing sweeps out foreign particles. Epithelial layer with damp mucous surface attaches bacteria. Lysozyme in saliva kills gram-positive bacteria. Phagocytic cells, polymorphs and macrophages. Slower flow rate of fluids over mucous membrane. Example, lysozyme complement interferon. Transferrin, lactoferrin virus inhibiting glycoproteins. Components of the immune system, example IgG, IgA, IgG, IgM, and lymphocytes and macrophages. Next point is normal flora prevent colonization of gut by foreign organisms, degrade biliary acids, use of essential nutrients. Next point is defenses of tissues, phagocytic cells. First point is phagocytic cells. Second point is serum components, antibacterial factors. Example, complement lysozyme, lactoferrin, transferrin, beta lysine, interferon, glycoproteins. Components of immune system, humoral and cell mediated immunity. Next point is, let's know about fourth point is general influences. Let's know about the general influences. Age and genetic factor. First point is age and genetic factors. Second is nutritional, endocrine and metabolic factors. Antimicrobial substances are found in sebaceous secretions. Triglycerides, free fatty acids and wax alcohols. Sweet gland secretions, lactic acid, amino acids, uric acid and ammonia. 5.5 pH of salvia also renders microbial effects. Next point, third point is substances derived from cornification process, steroids, amino acids, pentoges, phospholipids and complex polypeptides. Tears contain lysozyme active against gram-positive bacteria. Areas where there is a high flow rate of secretions such as the urinary tract, biliary tract and lower respiratory tract are generally sterile in contrast to areas where the flow rate is lower such as the oropharynx and the large bowel. Interference with flow rate in the urinary tract or biliary tract will lead to infection, nasal and salivary secretions. Now let's know about basic polypeptides. These have been isolated from leukocytes such as leukins, platelets such as plaquins, kidney such as spermine. This group also contains arginine and lysine container basic proteins, protamine and histone. The ability of these basic polypeptide is to destroy bacteria probably depends on the ability of the NH2 groups to react non-specifically with the Nissen acidic substances. Now let's know about beta lysine. It is relatively thermostable substance active against anthrax and related bacilli. Now let's know about the acidic substances. Now let's know about the acidic substances. It includes substances like lactic acid found in muscles and inflammatory zones. Humoral factors. Let's know about the humoral factors. In the blood, certain acute phase materials are present which increase in level in the inflammatory response. These substances may be C reactive proteins, alpha 1 antitrypsin, alpha 2 microglobulin, fibrinogen and celluloplasmin, and certain complement components, that means complex group of serum proteins that lyse RBC and destroy bacteria. The increase in the level of these proteins appears to be mediator that is released by phagocytes. Probably these compounds activate the complement. Now let's know about the interference. These substances are produced by cells stimulated by live or killed viruses and certain other inducers. Tissue and body secretions also contain other antiviral substances as inhibitors of viral hemagglutination and virus inactivating agent, that means VIA. In short, VIA. Milky also contains an antiviral substance. Now let's know about the normal bacterial flora. The normal bacterial flora of the skin may itself produce various antimicrobial substances such as bacteriocins and acids. At the same time, the normal flora compete with potential pathogens for essential nutrients. Certain other commensal microorganisms present in the body help in degrading the bile acids and stimulation of the immune system. Now let's know about the acquired or adaptive immunity. The resistance that an individual acquires during life is known as acquired immunity. The antigens of the invading microorganisms come in contact with the cells of the immune system, which leads to the initiation of an adaptive immune response specific for the inducing agent. The adaptive response improves with each successive encounter with the same pathogen. In effect, the adaptive immune system remembers the infectious agent and can prevent it from causing disease later. For example, diseases such as measles and diphtheria induce adaptive immune responses which generate a lifelong immunity following an infection. The two key features of the adaptive immune response are the specificity and memory. Now let's know about the active and passive immunity. Acquired immunity may be of two types, that is active and passive immunity. First is active immunity, the immunity that is conferred by a specific infection or by introducing an antigen through vaccination into the tissues is called active immunity. The actual material injected may be small quantities of living or weakened microbes. Example, polio vaccine. Small quantities of toxins or harmless antigenic materials derived from the microorganisms or in its toxin. This results in the active functioning of the person's immune apparatus, leading to the synthesis of antibodies and the production of immunologically active cells. Active immunity sets in only after a latent period which is required for the immunological machinery to be set in motion. Active immunity is of two types, that is lifetime immunity, second is short term immunity. Let's know about the lifetime immunity. It is caused by infections such as diphtheria, whooping cough, smallpox and mumps. Next is short-term immunity. Infections such as common cold, influenza, pneumococcal pneumonia and bacillary dysentery confer immunity for a shorter time. Next is active immunity may be natural or artificial. Natural active immunity results from either a clinical or in apparent infection with the parasite. A person who has recovered from an attack of smallpox develops natural active immunity. Artificial active immunity is the resistance induced by vaccines, which are prepared which are preparations of live or killed microorganisms or their products used for immunization. Let's know about some examples. First is smallpox vaccine was introduced by an English physician Edward Jenner in 1796. Second is another disease is Salk vaccine. Which vaccine? Salk vaccine against polio prepared by Jonas Salk in 1953 from killed polio virus. Sabin oral vaccine contains live but tamed virus, that is weak virus. Third is rabies. 14-day series of injections known as Pasteur treatment development by Louis Pasteur in 1885. Next is typhoid and cholera. TAB cholera vaccine containing Salmonella typhi, Salm, Paratyphi A and B and Vibrio cholerae. Initial dose 0.5 ml, second dose 1 ml. Now let's know about tuberculosis. BCG vaccine prepared from a culture of living bacteria named Bacilli calmette guerin after two French bacteriologists who made it. Next is diphtheria, pertussis, that means whooping cough, and tetanus DPT vaccine. In our country, the Hopkins Institute at Bombay, the Virus Institute at Pune, and Pasteur Institute at Kurnool produce several kinds of vaccines. Now let's know about the passive immunity. This type of immunity is attained simply by the transfer of actively formed antibodies from one person to another, the recipient thereby receiving preformed antibodies. There is no latent period in passive immunity, protection being effective immediately following passive immunization. This immunity is transient, that is not long lasting. It is less effective and provides an immunity inferior to that produced by active immunization. Now let's know about passive immunity. Passive immunity is of two types. First is natural passive immunity. Second is artificial passive immunity. Natural passive immunity is an important source of protection for the infant during the first few months of life, when his other antibody synthesizing capacity is relatively poor. The antibodies pass from the mother to the fetus during pregnancy across the placenta. Next point is artificial passive immunity is achieved by administration of antibodies. It involves the use of various animals, rabbit, sheep, horse, guinea pig, that may be infected and allowed to manufacture specific antibodies against some infectious agent. Blood serum of these animals containing the antibodies may then be used to protect man against the same infectious agent. Diphtheria antitoxin is an example of an IG when confers passive immunity. Similarly, pooled gamma globulin is given to a person suffering from certain infections such as measles, hepatitis, or tetanus. The protection afforded by this passive transfer of antibodies is relatively short lived, usually lasting only a few weeks. Finally, finished. Any questions regarding this, you can ask me in the comment box and subscribe my channel to know more updates. Thank you.